So hello everyone, and thank you for having me here. These are my conflict of interest. And let me start with some background information about EB research. EB was discovered over a century ago, and that was followed by the characterization of EB subtypes. The research in the last 50 years let us have an understanding of basement membrane structure and underlying defects in EB. Around 20 years ago, gene replacement attempts were initiated preclinically, and today these attempts continue with clinical trials. The common pathology in EB is a defect in attachment of basal keratinocytes to dermis, which leads to a separation in different levels of skin. Here, basal keratinocytes and basement membrane are shown in a schematic and immunoelectron microscopic image. The separation can occur within the basal keratinocyte if the proteins like keratin, BP230, or plankton are affected, and this results in EB simplex. Just beneath the basal keratinocytes lays basement membrane. This is a junction zone between epidermis and dermis, and the mutations affecting laminin 332, collagen 17, or alpha-6 beta-4 integrin would cause a separation in the upper part of basement membrane and result in junctional EB. At the inferior part of basement membrane, collagen 7 proteins form anchoring fibrils, which attach the lower part of basement membrane lamina densa to the dermis, and the mutations affecting collagen 7 causes a separation in the level of dermis and dystrophic epidermolysis bullosa. Gene replacement attempts were initiated on mouse models, such as this collagen 7 hypomorphic mouse. It expresses collagen 7 at about 10% of normal levels and resembles human dystrophic EB with blistering, nail dystrophies, and mitten deformities. However, one of the main problems of working on mice is the difference between human skin and mouse skin. Human skin has 6 to 10 layers of epidermis and a thick dermis, whereas a mouse skin has an epidermis of 3 layers and a faster turnover, a thinner dermis, and more hair follicles. Therefore, a mouse model, but including human skin, was needed to study this disease. That's why xenograft mouse models were developed. In this model, skin biopsies are taken from EB patients. The keratinocytes are separated from the rest of the biopsy. Gene transfer is performed using a retrovirus, and the corrected keratinocytes are grafted on immune-deficient mice. The graft is called a xenograft, and after a certain time, the graft is biopsied to assess the efficacy of treatment. This model was initially used for junctional EB. Collagen 17 or BP180 was selected as a target protein that is encoded by collagen 17A1 gene. Keratinocytes were collected from junctional EB patients carrying collagen 17A1 mutations, and they were corrected with a retroviral vector carrying the healthy collagen 17A1 gene. The corrected keratinocytes were grafted on immune-deficient mice, and the biopsies were performed. Here on the left panel, you can see the keratinocytes of junctional EB treated with BP180 or collagen 17A, and the epidermis that they formed is nicely attached to the underlying dermis, and also these treated keratinocytes express BP180 at the basement membrane. On the other hand, non-treated keratinocytes of junctional EB patients formed an epidermis, but this epidermis could not attach to the underlying dermis and blistered. Also, these keratinocytes did not express BP180 at the basement membrane. Another attempt in junctional EB was to replace laminin 332, or previously named as laminin 5. Laminin 332 deficiency was caused by mutations in 3 genes. And in the study, junctional EB patients carrying LAMB3 mutations were selected. Their keratinocytes were taken and treated using a retroviral vector carrying the healthy LAMB3 gene. The corrected keratinocytes then again grafted on immune-deficient mice, and biopsies were taken. The left images show normal skin, a healthy control showing healthy epidermis and dermis, and normal laminin beta-3 expression at basement membrane. On the right panel, you can see the junctional epidermolysis bullosa patients' keratinocytes treated with laminin beta-3, and the epidermis that they formed nicely attaching to the underlying dermis, and also the keratinocytes expressing laminin beta-3 at the basement membrane. On the other hand, junctional EB keratinocytes again, but not treated, formed an epidermis, but this epidermis blistered, could not attach to the dermis, and the cells did not express laminin beta-3. In the lower panel, it is shown that this gene treatment affected all layers of epidermis. And on the right panel, immunoelectron microscopic images of normal skin, non-treated graft and treated graft. In the treated graft, we can see the attachment of epidermis to dermis, and also formation of hemidesmosomal plaques, which are like junction zones or connection zones between epidermis and upper basement membrane. On the other hand, non-treated grafts of junctional EB patients show the detachment of epidermis and dermis, and in between a blister formation. Similar approaches were used for other mutations as well, for the correction of EB simplex cells with keratin 14 and junctional EB cells with beta-4 integrin and alpha-3 chain of laminin 332. Recently, the research in this field is focused on recessive dystrophic EB, which is caused by loss of type 7 collagen due to COL7A1 mutations. Type 7 collagen is a large protein. It has a triple helical collagenous domain flanked by non-collagenous parts NC1 and NC2. Multiple collagen 7 proteins assemble to form anchoring fibrils, which are important in attaching basement membrane to the underlying dermis. Similar to the collagen 7 protein itself, the gene encoding collagen 7, COL7A1, is a large gene which posed technical difficulties in transferring it into a virus. In addition, there was a discussion that large capacity viral vectors might have oncogenic potential. Therefore, initially, non-viral transfer methods were tried. One successful approach was using a plasmid to carry collagen 7A1 gene. Here we see an example. On the upper panel, RDB keratinocytes without treatment. On the middle level, in the middle layer, RDB keratinocytes with plasmid using with treatment using a plasmid carrying collagen 7A1, and in the lower level, normal keratinocytes for comparison. And you can see the treated keratinocytes expressing collagen 7 in immune fluorescence. Also, similarly here, treated keratinocytes forming an epidermis, attaching dermis, and expressing type 7 collagen at the basement membrane, whereas non-treated keratinocytes formed an epidermis but could not attach to dermis and blistered, and those cells did not express type 7 collagen. Although that was a successful approach, the effects did not last long. And viruses has been demonstrated to be more efficient in transferring genes into human keratinocytes. At some point, FDA approved the use of retroviral vectors in EB research, and this is one of the vectors designed to carry collagen 7A1 for the preclinical trials. The same approach was followed. Skin biopsies were taken from RDB patients. Their keratinocytes were separated from the remaining tissue, and transduced, or the missing gene, collagen 7A1, was transferred using this retroviral vector. The corrected keratinocytes were grafted on immune deficient mice, and the xenograft was biopsied after the transfer, the treatment, to assess efficacy. Here on the upper panel, you can see the RDB keratinocytes without treatment, and on the lower panel, RDB keratinocytes with gene treatment expressing intracellular collagen 7 protein and immune fluorescence examination. Also in Western blot, the whole length collagen 7 expression was detected in the treated mice. One of the highlights of the study was showing the durability of type 7 collagen expression. The engrafted cells were tracked over 12 months using a bioluminescence measurement, and 12 months after the grafting, the bioluminescence signals were still present in the grafts. Consistent with this, type 7 collagen expression persisted throughout the experiment. The study also showed the importance of transducing the stem cells. Normal epidermis undergoes self-renewal every 4 weeks. New keratinocytes are produced by stem cells which reside in the basal layer of epidermis. To achieve an efficient and durable gene treatment, epidermal stem cells should be targeted, and the number of treated stem cells should be maximized because naturally a competition occurs between treated and non-treated stem cells. Therefore, monitoring the stem cells seemed to be an important factor. Following this preclinical work, FDA approved conducting clinical trials on RDB patients using the same retroviral vector. The same approach was used. Epidermal sheets were constructed for the patient using their own keratinocytes and grafted on their open wounds. Phase 1 and 2 trials have been completed and the results have been published. Phase 3 trial is still ongoing. A different attempt to supply collagen 7 to RDB patients was using fibroblasts. Fibroblasts are located in the dermis of skin, and they also produce collagen 7 in healthy individuals. The preclinical study was conducted using RDB patient skin. Fibroblasts were separated from the rest of the tissue and treated with collagen 7A1 gene using a lentiviral vector. On the other hand, full-thickness skin grafts were prepared again using the patient cells and grafted on immune-deficient mice. And the treated fibroblasts were this time injected into the xenograft so that the fibroblasts could go to their exact location to the dermis. The biopsies were taken and they showed collagen 7 expression at basement membrane in the grafts treated with fibroblasts, collagen 7A1 carrying fibroblasts. And also these grafts did not show a detachment or blister formation as opposed to non-treated fibroblasts or grafts injected with non-treated fibroblasts. On the right panel, you can see the treated fibroblasts expression collagen 7 compared to non-treated fibroblasts. Another group also showed the gene transfer using fibroblasts with collagen 7 expression at basement membrane, and in immunofluorescence examination, they showed the formation of anchoring fibrils or the beginning of formation of beginning or initial anchoring fibrils extending from the basement membrane to the dermis. After the preclinical studies, phase 1-2 clinical trial for fibroblast gene therapy has been completed as well. The phase 3 study is currently enrolling patients. So far, I have spoken about ex vivo gene treatment, where patient cells are taken, treated in the lab, and given to the mice as a graft or subepidermal injections. Another approach is in vivo gene treatment, where the gene product can be directly administered. To study this, RDB keratinocytes and fibroblasts were taken from patients and grafted on mice. Then the gene product or the vector carrying the gene product was applied directly on the xenograft. This system used a herpes virus vector, which is big enough to carry collagen 7A1 gene. After correction of the RDB cells, collagen 7 expression was shown in keratinocytes as well as in fibroblasts. The biopsies from the grafts show collagen 7 expression at the basement membrane with NC1 and NC2 domains, as opposed to the control grafts where there was no collagen 7 was expressed, and also the control grafts blistered. Finally, immunoelectron microscopy showed NC1 and NC2 domains of collagen 7, NC1 exactly here, and NC2 in the dermis, and also the anchoring fibril formation, the beginning of anchoring fibril formation between the two domains of collagen 7. In summary, gene replacement using viral vectors was preclinically successful. Genetic material could be transferred ex vivo and in vivo, and clinical trials were conducted in junctional EB and RDB patients. Thank you.